Hey guys, so I decided because of the snowpocalypse outside that it might be best if we do another podcast because I don't know when we're going to see each other again. And the end of course exam does not get postponed because we've had bad weather. That's a state mandated exam, so the date that they've selected is the date they've selected, like no matter what. So, anyway, it's time to start genetics, so here we go. Okay, this is Gregor Mendel. He is an Austrian monk who is the father of genetics. Many of you may not know what a monk is. Okay, it's someone who's devoted their lives to the church and ministry and sort of things like that. It's kind of the male version of a nun. Back in the day, they were really strict, and so monks cannot get married. Monks sometimes take a vow of silence. Monks have to grow all of their own food, take care of their church, scrub, clean. So they have to be completely self-sufficient within their church because they all live there. And they make, no money except to upkeep it from donations from people who attend the church. But generally, places where monks live are private. So anyway, Gregor Mendel is the father of genetics, and he spent a lot of time growing peas in the garden. And so that's where we actually began our genetic journey. Okay, so here's a picture of pea plants because a lot of you know what a pea is, but you've never seen a pea plant. So here are the types of things he worked with. If you've ever heard the saying, you're like two peas in a pod, well, this down here would be an example of a pod, and inside each pod is between 2 and usually 3 peas. Here are the blossoms. They're typically purple in nature, but can also be white. This is a tall pea plant, and this is a short or dwarf pea plant. We're going to be talking a lot more about these, hopefully when I see you soon. Okay, so here are all the traits that he studied in that you really can study with pea plants, at least the ones he used. So you can study flower color, which we talked about before is purple versus white. Flower position. Axial is just a term that means the flowers are on one of the bottom layers. And then terminal means that they bloom from the top. Okay? As seed color, your options are yellow and green, and the shape can either be smooth and round, which is what most of you eat, I think, and then your other option is wrinkled, which they kind of look, I'm not gonna lie, they kind of look a little gross when they're wrinkled, which is probably why we sell smooth peas. They're more consumer friendly. Okay, and then you've got pod shape, you've got the inflated, that just means it's big and like puffy, like as if you were to blow up an inner tube with air. It kind of looks like that. And then you have the constricted, which means that the bumps you can see are each of the peas within the pod. Pod color can either be green or yellow. And then stem length, if you can see, let me scoot that down. Stem length is either going to be tall or dwarf, okay? We'll get into why we care about this in a second. Okay, here are Punnett squares. These are things that you've probably done 1,000 times in the 8th grade, but it never hurts to go over these. There are tons of practice ones in your packet, and I want you guys to go ahead and do that very first page of Punnett squares, because I think it's like SpongeBob's Bikini Genetics or something. I'm sure a lot of you have already done it, but it's just an initial practice to get you back in the habit of how Punnett squares work. So I'm going to go over it with you once right now, and then I'm going to post the key on Edline to that first SpongeBob's Bikini Genetics. Probably, what day is it? I'll probably post those Thursday. That'll give you guys enough time to have fun with the snow and still be able to learn something this week. Okay, so let's go over the squares so that you know that you're doing it right, okay? So up here you would put, let's say, the maternal genes, okay? That means for your mother or the mother plant, the female plant. And then let's say that we're going to put the dad's genes down here, or the male donation, the male plant. Okay, so what happens is you take, to fill each square, let me see if I can get there it is. You take one letter and put it in the box, and then you take the corresponding letter and put it in the box, okay? And then same thing with this square, you take the top letter and put it in the box, and the same letter over here and put it in this box. It's kind of like foiling if you've ever done that in algebra, except this is way easier. Okay, now let's do one where it gets a little trickier. Let me see if I can find one. This one's a good one. This next one over here. Okay, so we're going to take you always do the capital T first. Okay, so because these are both capital, it doesn't matter which one goes here and which one goes here, but you always put the capital letter first. So this is what we call homozygous dominant. Homo just means the same, and then, zygote or zygous. So what we're looking for is, in homozygous, we are looking for two letters that are of the same capitalization or lowercase. And so here we have homozygous dominant because anytime you use a capital letter, that is a sign for dominance in genetics. So same thing with this next box, because they're both capital, it doesn't matter which one you put first. We have another homozygous dominant here, and I promise we'll go over this again. And then, okay, here's the deal. When you get to the bottom box, okay, remember, you take the top letter from the mother and put it in because it's capital, so it gets to go first. And then the lowercase from the dad, and this is heterozygous. Hetero just meaning different, okay? And then you do the same thing over here because this is a dominant. You put its capital letter first, and then the recessive letter from the dad over here. And you get another heterozygous. So these are all four chances that the four possibilities that could come from that combination, okay? So in this combo here, let's go back. This family would have a 50% chance of having a homozygous dominant child every time for that trait. And then they have a 50% chance of having a heterozygous for that trait. And we'll get into phenotypes in just a second. Okay, so let's talk about monohybrid crosses. You guys probably should write this down, at least in your own Cliff's Notes version, because you're going to have to study for the test based on these notes. So monohybrid cross is a cross between parents who are true breeding, and what that means is like purebred, okay? So the first cross has to be between two purebred parents. So it's I don't know how to explain it. It's kind of like with dogs. In order to get a golden retriever as a child, as a puppy, you have to have two purebred golden retriever parents, So that's the same thing with plants and all things. So a monohybrid cross is a cross between parents who are true breeding or purebred for a trait. And what that means is that, if you look at my little example here on A, both are homozygous, okay, for one allele of the gene. And all that means is they're either both capital A, or they're both lowercase, lowercase. Either way, they have to be homozygous for that gene, okay? And the typical cross is you do a homozygous dominant and you cross it with a homozygous recessive so that you can see what traits are dominant and what traits are recessive. Let me see if I can find. So capital A, as we've talked about, is dominant allele for a trait. And lowercase a is the recessive allele for the same trait. So let's see. I've got some more crosses here. Here we go. So that's a monohybrid. Now we can get into dihybrid crosses, which get a little more complicated. I think in the 8th grade you guys may have done monohybrid crosses, but you shouldn't have probably done dihybrid crosses because they're pretty complex. Not that you guys couldn't handle it. I don't think it's in the 8th grade curriculum, if I remember when I taught 8th grade. Okay, so this is a cross between their offspring. Okay, so once the monohybrid crosses are done, you can then cross their offspring, for plants. And so this is the dihybrid cross, which parents are both heterozygous, which means, one capital, one lowercase, at one location. So if you're looking at our dihybrid cross down here, okay, in this example, they're doing it for peas, And it seems to me like they're doing yellow versus green. And smooth versus wrinkled is what that looks like. Okay, so if you're looking, hang on, let me get my thing working. Okay, so they've done the paternal plant crosses on top and the maternal down the side. It honestly doesn't matter. It doesn't matter if you want to put the maternal on the top row or on the side row. It doesn't matter. As long as you know when you're crossing that the dominant letters are the capital letters go first. That's all you need to know. This is something that I would rather practice with you in class, but if I absolutely know I'm not going to see you, we'll come back to this lecture and I'll have like a dry erase board and we'll do it together, okay? Oh, Mendel, of course, Mendel, I remember you. Yes, and we remember Punnett squares. Okay, so now we have to talk about some other types of dominants other than just regular dominant versus recessive. Things get more complicated. If you are just initially watching this podcast, I would ask that you stop here because you're going to get confused. So if you could stop what you're doing here and go practice that SpongeBob so that you can check online on Edline and see if you're doing it right, and then email me if you're not, okay? And we'll put another video up here. What I'm about to say next is it's more in depth and it's gonna confuse you. So stop here and I'm gonna post another video talking about incomplete dominance. If you have already done a lot of this and you feel more advanced, go ahead and do that SpongeBob thing anyway and then pick right back up with this next podcast, okay? Thanks, guys. Hopefully I'll see you soon. Stay warm.